Alright guys, so I do apologize for it taking so long to get the bubble lab up there, but I was waiting for the students to do it in class so I could video it and then show you exactly what had happened. And it took us a little bit longer in class to do the bubble lab than I thought, with the classes being shortened. So anyways, we have this bubble lab, and it's basically modeling how our cell membrane is. So you guys hopefully have seen that by now. So you also have where you're supposed to draw this. And I wanted you guys to realize in Biology 1, we see it like the outside of the cell, and we're like, oh, that's the cell membrane. It lets things come in and out. But we don't realize in Biology 1 is what it truly looks like. And it's not just this line that we draw on a paper. It's this fluid mosaic. It's got this double layer, and you can see where I'm circling here. There's one layer on top, and then there's this layer on bottom of phospholipids, where the heads are water-loving, they're hydrophilic, and the tails that are pointing inward are hydrophobic. And there's all these proteins all over the place, and you can see these channels where things can get in and out of the cell. Now, some things can pass right through the phospholipid bilayer, but some things need help, and they need these channels because they're a little bit larger. And it doesn't mean that everything can pass through. There's some channels that you have to have a certain fit. But then if you look, there's these carbohydrates out here, and sometimes those are used in cell signaling, because no, cells don't talk, but they do signal each other and can, it's almost like their own little language that they have. So you've got signaling, you've got cholesterol in here, there's all kinds of different things going on within the cell membrane. And it may seem like it's not that important, that, oh, it's just the cell membrane, but we do have, when it becomes important is when it affects us as humans, and we have diseases, and cystic fibrosis being one of those big ones we'll talk about here. So all of this needs to work correctly. We need to know about this so that we can help people out that have illnesses. So here is just a single piece of this phospholipid bilayer. It's just one. I talked about this head that they are water loving, and then these tails that are water fearing. And you can see this right here. So the water would be out here. Like if this were the outside of the cell, this would be out in your body. This would be inside the cell where there's a lot of fluid. So the proteins I was talking about, I said that there are some that have channels. There are some that are very specific to shape. So we have channel proteins, and these are just small openings for molecules to diffuse through. So there are molecules that can't just fit through the phospholipid bilayer, but they can fit through these small openings. Now, carrier proteins, there's a binding site, so it grabs only certain molecules and will help them through. And then we have gated channels, which are very similar to the carrier proteins, and they're not always open. So only certain things can come through, but they're not always going to be open. Like if you look here at, let me see here, this protein right here, you can see where if something comes and sits right here, it will cause it to open up. Or this one right here, not always necessarily open, but this one is open. So is this one. That's a channel protein though. All right, cholesterol. Cholesterol stiffens the membrane by connecting these phospholipids. And then we have some of these carbohydrates that are throughout here. And glycolipids, those are signal molecules. Like I said, they can send signals to each other. We don't necessarily think of ourselves as being like their own little living entity, but they can signal each other. And then these glycoproteins, super important for us. Once again, it may seem like, why do we have a whole section devoted to the cell membrane? This is why. If we're talking about like right now in the time of COVID, these are where our antibodies are going to be. So if you have COVID antibodies, they're going to be on your cell membrane out here. And if you see that molecule, that invader in your body, that cell's going to recognize it right away, and it's going to neutralize it. All right, so here is HIV and how this works. So HIV attaches to a receptor. So it attaches to one of these receptors and kind of tricks our cell into letting it come in. So it attaches to the receptor right here. The cell thinks that it's something it should let into the cell, and it does that. And if you look, this is really the problem, is it's DNA. So once it releases these DNA particles in here, it then incorporates its DNA into ours. Then our own nucleus and DNA and ribosome start making more of the virus. that eventually gets put back together. We'll have a whole coloring page that breaks down how HIV works a little bit more in detail than what I just went into there, because that's a very, very quick summary for just this one slide. All right, and then we have, we have something called passive transport, and that means that our membrane is selective permeable. It allows certain things to come, oh my goodness, I'm sorry. It allows certain things to come in and out. So if you look at this is a non-charged molecule, and you see it, it's going in and out, back and forth. And then you've got this larger macromolecule, probably it looks like a protein of some sort. It's too large to fit through. So you can see where it's going to bounce out. Water can flow in and out of this cell, so you can see that. But then charged molecules and ions, they will bounce back and forth. They need some type of channel to help them out. So that's the things that can pass through, the things that cannot pass through. Passive transport, that requires no energy. That's what passive means, right? Passive versus active. If you're active, you are actively using energy. Passive, no energy, it just happens. So this is simple diffusion, water, oxygen, these small molecules that can just go in and out as they please, and it's always from an area of high concentration to low concentration. I know I've said this in other, Biology 1. If you remember back that far, I talked about how if I were to spray a perfume in the corner of the room, I would be like, who's the first kid that's going to smell it? And it's usually the closest person to me. The person furthest away is going to be the last to smell it, because it's going from an area of high concentration in that bottle, and it will spread out into an area of low concentration until eventually maybe you just can't smell it that well because it's so low concentrated. And they're showing that here with diffusion. We see this when we, if I were to get a beaker of water and put a drop of food coloring in there, you would see how it'd be concentrated and then it just kind of spreads. All right, facilitated transport or diffusion. It's diffusion, things are moving from an area of high concentration to low concentration, but it's facilitated, so it needs a little extra help. So it's got a protein that kind of helps it out here. So we see diffusion where it doesn't need the protein, but this one, there's a protein. Maybe they're just a little too big to get through there. They're still going from areas of high concentration over to areas of lower concentration, but it needs that protein. All right, carrier proteins are just that. It's almost like they're carrying them through. So it's not going to let everything through. So these things cannot go across the membrane. So if you look, these little like hexagon looking guys, they are not going to fit through here. When this little circular oval shaped one hits right here, this carrier protein will open up and carry it over into the other side. All right, so here is something, if you remember back to Biology 1, we did the gummy bear lab. We talked about hypertonic, isotonic, and if you remember those gummy bears, the saltwater bear became smaller and a little bit harder, and then the others almost got like watery. If you were lucky enough to keep yours together, they were very just slimy and they fell apart very easily, but they were a lot bigger. They had really absorbed a lot of water. So why would you not want to use distilled water in an IV bag? See, an IV bag here, it's got a mixture of all these salts and everything in it. If you used pure distilled water, that pure water that's a very high concentration. So when it gets into your body and sees your cells are a low concentration. So that water is going to flow from the high concentration that was in the bag right into your cells. And it's going to be hypotonic. Your cells are really going to swell up, which is not good for you. You don't want to be dehydrated, but you also don't want them to swell up like that. So that's why you would not want to use distilled water in an IV bag. Our cells don't, but there are some animal cells that have something called a contractile vacuole, and they pump out fresh water. So you see this in a lot of our animal-like protists that are in pond water. They're in water, so of course they're surrounded by water, so they need a way to pump water out, and that's what this contractile vacuole does. There's one right here. You can see where water comes in. This contractile vacuole gets a little bit larger, and eventually it expels the water out. Now, I'm going to go back a slide real quick to right here. Our cells have just a cell membrane and water can flow in and out very easily. Now a plant cell is going to look a little different because they have the cell wall. So hypertonic, I think of that hyper little kid and your cell's going to get smaller. Well, the cell wall does not shrink, but the membrane does. Isotonic, everything's fine, everything's normal. And then you have hypotonic, and you can see where the cell membrane is pushing against that cell wall. Now this is called turgor pressure. So when it's pushing, think of pressure. Pressure is when you push, when there's some pressure to something. So you have a lot of pressure on that cell wall in the hypotonic. If you look here, there's no pressure, really no pressure at all, and it's called plasmolyzed. And then if it's just normal like this, it's called flaccid. But there's not, you can see there's not pressure on that cell wall. All right. So same thing in U-tubes. There's this thing called a U-tube, no, not the channel online. But what happens is if you look at this, you can put water in this tube and it's got this permeable, selective permeable membrane right here, just like our cells have. And if you add like salt or something to this side, well, when you add that stuff to this side, all of a sudden, look, the water, there's not as much water compared to this side, right? Like the concentration is not as much. So we're gonna go from a concentration of high to low, and you can actually see where the water will kind of just seep over into this other side, and you get this difference then. So it's kind of, it's just watching osmosis in action. All right, 5.3. Now, at any time you can stop. Each day I will tell you exactly what slide number we got to. Right now we are on slide 16. I'm doing this before I do it with the class, so I'm going to assume that there's no chance that we will get this far within the first day. And like I said, I will let you guys know that. I'll post this video, and I'll let you know each day exactly how far we got. I don't expect you guys to do all of the notes all in one sitting. So just keep that in mind, and I will let you know each day. All right, so 5.3, let's see where we're at here. 5.3 is active transport. Now, we just did passive transport. That required no energy. Now, active transport is going to require energy, and the simple fact, the only reason that is the case is because it is uphill. So normally, every time we are going from an area of high concentration to low concentration. So this is going to be opposite. We're in low concentration area, and we're going to push it uphill to the high concentration. And that's going to require energy on our part. So the types of passive transport that we've already discussed, well, we saw you have diffusion, you have facilitated diffusion, you have these carrier proteins that we saw back here, all part of passive transport. So there's a lot, and we'll have a worksheet that summarizes all of this in one chart. So we'll have that, and it'll be good to kind of look back at this and use these notes to help you answer those. All right, so one form of active transport is going to be a sodium-potassium pump, and guess what? It pumps sodium and potassium. So if you look at this model here that it's showing, you get 3 sodiums and then 2 potassiums. So you're pumping out sodiums and potassiums, and this little phosphate right here, you see it coming in and out, that little phosphate? That is part of our ATP, which is our energy. That is a phosphate because ATP is adenine triphosphate. We have phosphorus in there. These phosphorus, that's our energy. So a huge amount of energy in our bodies is used to power this pump and prevent sodium from building up in our cells, right? That's not going to be a good thing. If we had too much sodium in our cells, could you imagine first off, you would be really thirsty, you would be dehydrated. So that's not a good thing. We need to get that pumped out, and we need to get it, usually it leaves in waste through urine. All right, here is a CFTR channel. A mutation in this channel protein prevents the movement of ions across the membrane. Mucus becomes thick and not enough water, leading to symptoms of cystic fibrosis. Now, I'm not going to go in too much depth on this one for the simple fact that we will do a case study on cystic fibrosis, and we will look at all the different things that can happen to this channel to cause it to not work, and we can also look at the drugs that have helped. And we can also look at the certain cystic fibrosis cases where the drugs cannot help, just depending on the genetics. So I'm not going to go into a bunch of things here, but if you look, you can see that this pore is clogged. What is a pore? It does not look normal. It's not a normal pore. So it gets clogged up, and when you get that mucus where it feels like you have to clear your throat. Now we can get that when we're, maybe we have a sinus infection or something like that, but this is, healthy people that don't have a sinus infection. They just deal with this all the time because their cells are that way. A co-transport, that is just that. Think of a co-captain, co-teacher, anything. You've got two. So what happens is this little protein right here, it will not flip over like this until both of these are in here. You have to have this orange and two of these red in order to get this protein to flip. If you just have an orange one sitting there, it's going to sit there for a long time. So co-transport, you need both of these molecules in order to flip through there. And if you look, you have some glucose and some sodium. And once again, it's going to cross a concentration gradient. All right. Oh, sorry. The next one is bulk transport. And we have endocytosis, and we have something also called exocytosis. But endo means inside, it's coming into the cell. And now you can bring in food particles, which is called phagocytosis. Those are the solids that your cell brings in. Or if it's water that it's bringing into the cell, then it's pinocytosis. So it shows an example of this right here, bringing this in, and then there's an actual microscope image of an amoeba engulfing a bacterial cell. So it's kind of hard to see, but this right here is the amoeba, and the bacteria is right in here. So it's kind of hard to tell what's going on there, but that is what's happening. All right, here's another image. There's an amoeba, and it is just engulfing, and our cells do something very similar to that. Not quite like this, but it's similar. And now it's stuck inside the amoeba, which is an animal-like protist. All right. Here's another one. It's a receptor-mediated endocytosis. So normally endocytosis will just happen. Your cell membrane will fold in on itself and pinch off. Now this one you have to have certain shapes. Do you see this? It's all shaped. To me it's almost like a puzzle. So the circles bounce around in here, nothing's going to happen. These triangles come in. And now this is very simple to show you guys what's going on. We know that a lot of our chemicals are not triangular shaped or anything like that, but you get the point. So they come in here and they fit into these receptors, and then that triggers our membrane to fold in on itself and bring these substances in. All right, so clathrin is a protein that stabilizes the membrane and pulls it inward to surround a particle. So you see, watch this right here. There's receptor proteins, and it pulls that in right there, and then it will, here we go, surround it, and then now it is inside your cell. So watch that again. There's the receptor protein. You can give it a second. And the clathrin is all this right here, but it helps. So you can see the little red things right here, the clathrin. And it's just a protein that stabilizes. It helps stabilizing that membrane so this can happen. All right, so exocytosis, exact opposite of that. You're exiting the cell, releasing, so you'll have these bubbles and these vesicles that we've seen through our cell unit, and it will attach to the cell membrane. You can see it kind of happening in steps here, and then it just kind of releases it open, and then this just becomes more of your cell membrane. So if it's pinching off and coming into your cell for endocytosis, you're like, oh, you're losing some of your cell membrane. Well, here you're gaining some back. All right, so mini labs and demos. We did the bubble lab. If we do any more of these, I will let you guys know, and I'll do videos and pictures and let you guys see those on Google Classroom. If you guys have any questions, let me know. See you later.